Welcome back, guys. Okay, today what we're going to talk about is a simple review that will help you for this year's final. What we've been talking about the entire year is obviously biology, and remember that all living things are made up of cells. Okay, and the most important concept that you need to take this year is that these cells, in order for them to stay alive, they need proteins, okay? And proteins are tools that the cell needs to stay alive, to keep it alive, okay? And so without proteins, the cells would die because it wouldn't have all the tools it needs, okay, to make energy, to maintain its DNA, to maintain its cell membrane. Proteins are the tools that the cell needs to do every single job inside the cell, all right? Now, how does a cell make its proteins? That is the key. That is the idea that you need to understand, okay? How does the cell make its proteins, all right? Now, the first concept is, well, the first step is cells have DNA, okay? So cells have their DNA. Every single cell has DNA. It doesn't matter, bacteria, animal cells, human cells, any type of cell is gonna have DNA. Where did this DNA come from? Okay, from its parents, from the other cells, okay? So in our case, our parents gave us our DNA, and that's the DNA that's inside our cells, all right? So cells need DNA. Why do they need DNA? Because DNA has all the codes, okay, necessary to build the proteins, okay? And these codes are, the fancy word for that is genes, all right? Or another way you can think of genes are as protein recipes, all right? So all our cells in their nucleus have DNA, and this DNA holds all the codes that the cell needs to make its proteins, okay? Now, how do we get to the protein? How does the cell make the protein? Well, we got to start with the DNA, right? Now, let's imagine that we have this code, this gene, all right? Now, this gene holds, is a code that will that has information to make a particular protein, all right? And there are many genes that DNA has, all right? Maybe 20,000 or so. Now, what the first thing that needs to happen is that this code, right, needs to get copied, all right? And it needs to get copied in a process called transcription, all right? Because remember, DNA is in the nucleus, all right? And the guys that are gonna make the protein, okay, are called ribosomes, okay? So the ribosomes are literally, okay, in here, right? Out here in the cytoplasm, right? And on the rough ER, but DNA is inside here. So we gotta take the information and send it out to the ribosome, okay? And that's why we have to take this code and copy it, all right? And copy it and turn it into a different version, okay? And that version is called mRNA, right? So during transcription, the gene gets turned into mRNA. How does this happen? Well, it uses the base pairing jewels. So if there's a guanine, okay, then there's an enzyme that adds a cytosine. Thymine, adenine, cytosine, guanine, G, C. A's goes with U, right? So because remember, we're making RNA, it wouldn't be a T. And so once transcription is over, now we have a copy of the gene, but in RNA form, all right? Now that this copy of the gene is ready, it goes to the ribosome, right? And the ribosome is going to use, okay, its knowledge of the codes, right? And in this process called translation, right, it's going to read the information 3 codons at a time, or 1 codon at a time, right? And each, oops, messed up here, right? So 3 nucleotides at a time, right? And if you take a look, CAG, that means that that's glutamine, right? So the first amino acid in the protein would be glutamine. Okay, CUG, that would be CUG, that would be leucine, and the last code, CAG, is the same as the first, so therefore this protein, the ribosome, is going to build a protein that is 3 amino acids long, and this protein would go, once it's built, it would go to wherever it's needed and do a specific job, all right? And that's how a cell makes all its proteins. Okay, what's the difference? Remember, this would be one gene. Okay, a different gene would have different codes. Therefore, the ribosome would read it differently and connect different amino acids. But it's that simple. Cells have DNA. DNA has these things called genes. Okay, the genes need to get copied to mRNA during a process called transcription, right? So that this copy can go out to the ribosome, okay? And the ribosome reads this information, okay? Understands the codes and uses the codes to figure out, okay, I need this amino acid, this amino acid, this amino acid, and it connects them together to form a protein. And once this protein is formed, it goes and it does a very specific job inside the cell. All right? Hope this helps. Good luck on your final this year.